Okay, so in this video, we're going to be talking about how to design an experiment to test a hypothesis, or basically the basic premise of experimental design. So many questions that experiments are designed to answer often start with a simple observation. And so for the purposes of this video, we're going to be referring to observation as simply watching or visually collecting information, or it's a process that could involve very careful measurements taken with very complicated devices. So we'll use the experiment that was introduced in the reading that we had, where somebody planted 10 beans in a pot, put it in a window, and made the observation that none of the beans grew. They also made the observation that they forgot to water the beans. And so this leads to the question, do these beans need water, these seeds need water in order to germinate? And so this will lead us to the creation of a hypothesis that seeds do need water in order to sprout or germinate. And for the purposes of this, and the best definition that I've found in my searching of what a hypothesis is, we'll define a hypothesis as a tentative answer to a well-formed question. And so the question again is, do the seeds need water in order to germinate? And the hypothesis is the seeds need water in order to germinate. The important part of a hypothesis is that it's framed in a way so that it's falsifiable. It's something that can be proved wrong by experimentation. And if you go and take a look back at that reading, they go into really neat examples of how some hypotheses are not actually falsifiable, and so they need to be redefined if you want to address them experimentally. Okay, so when you're designing an experiment, you're always going to want to include a control group or many control groups in your experiment, in addition to an experimental group. And one thing that's really important to remember is when you create these groups, you need to control all of the variables in the experiment. So what are we talking about when we're talking about variables? Variables are anything that can change. It could be the type of soil, the amount, the duration of sunlight, the type of pot, the size of the pot. All of these things are variables, and it's absolutely important that we control each of these variables between the different groups that we have set up in our experiment. We are going to control all these variables except for one variable that is allowed to change. In this experiment, it's going to be the water. Our control group is going to have no water, and our experimental group is going to be exposed to water. And this variable that we're changing, the water, is actually called the independent variable. And the independent variable in any experiment is the variable that the scientist is testing, and it's the variable that the scientist is controlling and changing between the two groups. If we do in fact end up seeing a difference between these two groups, and we accidentally planted them in two different types of soil, so we had two different variables changing, water and soil type, we would not be able to tell if the difference that we see between the two groups is due to the water or due to the soil. And that's why it's so important to only change one variable in a given experiment or at a given time. Okay. So then we're going to make some observations later on, and we're actually going to take some data, so collect data in the experiment to find out the outcome of our experiment. In this case, we're going to look and see how many of the seeds sprouted or germinated in each pot, and we're going to come up with a germination rate for each pot. So 9 out of 10 in the plus water group, the experimental group, and 0 out of 10 in the minus water control group. Okay, and so that's actually going to be the data that we're collecting. That observation is the data that we're going to be using. And so once we have that data, it's really important that we can communicate that data to other individuals and other scientists. And so one great way to do that is to visually display the data. And so you probably have seen graphs or charts that look something like this. Many people call this a bar graph. The program Excel actually calls it a column chart. And one feature of it that is going to be kind of consistent between different types of experimental data you look at is that oftentimes the independent variable that the scientist is testing is going to be on the x-axis, and the dependent variable, which is the variable that you're collecting. So it's the data that you're actually collecting that's dependent on the independent variable, is going to be on that y-axis. You can see here that the two different categories case, on the x-axis are those independent variables, and you also have a label for the x-axis in addition to the category labels. And on the y-axis, you have a description of what you're actually seeing in addition to the units, which in this case are actually percentage, because it's a ratio. Oftentimes when you display a graph like this, you're also going to have what's known as a figure legend located below it. The figure legend is going to be just a very brief description of what you're actually seeing in the graph itself, and it kind of supports the visual data in the graph and kind of fills in some of the gaps, explaining what happened in that experiment. It's often going to have some identifier like figure 1 or something like that, and a title that's descriptive yet simple, and kind of tells you what's going on in that experiment. And we'll talk more about figure legends later on. Okay, so this is all well and good, but if you've noticed in our experiment, we really only had one experimental setup or one data point, the 9 out of 10 and the 0 out of 10 for each of our groups. And so one really important feature of experimentation is repetition, and this is to ensure the accuracy of our findings. We really should have multiple trials or subjects for each group. And so if we were to do repetition in this experiment, we might have multiple pots for both the control and the experimental groups to get that repetition. And this is going to be really good because it improves our accuracy of our findings. And it also allows us to display, instead of an individual data point, it allows us to display the average of each of our groups, of our experimental group and our control group. Once we have that average, we can also do other things like plot error bars showing standard deviation or standard error of the mean. And those are going to be really important because they can help us understand a little bit more about the individual data points within a given group. It allows us to see things like dispersion and other things like that within a group. Just so you know, there's going to be an entire another video that's dedicated to talking about standard deviation and other statistical analysis. And there's also a separate video that's dedicated to walking us through the process of plotting data in Excel. Okay, so in addition to the standard deviation and things like that that I had just mentioned, the other really important point about repetition is that once we have multiple subjects within a group, we can actually use those data points to find statistical significance between the two different groups. So if you do see a difference between the two different groups, you can apply statistical analysis in order to determine if there is a statistical significance in that difference between the two different groups. And so that's another really important reason to do repetition within an experiment. And one thing that I wanted to just mention after we've kind of talked about the experimental process is that we talk a lot about the scientific method or this process that we use to answer questions about observations in the natural world. And it often seems like a very linear process. And in fact, it's actually much more complicated than that. Science often is a process where we'll start off with one experimental design and start to generate data, and preliminary analysis will send us right back to the drawing board, make us realize that we need to change our experimental design in some way to account for variables that weren't obvious at the onset and other factors like that. And there's a lot of circular things that happen in the process of science. There's also a lot of influence and influencers in science. So science, by its nature, is going to exist to serve society in answering some questions and spurring the development of certain technologies and things like that. And because of that, actually, there's a lot of different factors that can influence what type of science that scientists can ask, that they're allowed to ask legally, so different types of legislation, and also what they are funded to ask, so what governments and other types of funding groups determine to be significant and important questions to investigate. And so that's going to come into play in the type of science that scientists can do. Another thing to remember is that science will often generate many more questions than it can answer, and some of the best scientific studies often leave many more questions than they've answered. Another really important one is that science does not happen in a bubble. The scientific community is going to guide the process at every step of the way, past literature, collaborations between other individuals in an institution, outside of institutions, across the globe collaboration. This is really important to make sure that the science is as good and as rigorous as it can be. Another aspect of that scientific community is that no science is ever going to be published in a reputable journal unless it goes through the process of scientific review first. And so that review, often called peer review, is a process by which the data from an experiment, after it's been written up and submitted to a journal, is reviewed by multiple experts in a field. And it's reviewed for all sorts of reasons, and most importantly, the process of the science is reviewed. Data collection, experimental design, all of these things are reviewed to make sure that the science is rigorous and sound before it can be published and accepted by the scientific community. And that process is extremely important, and it can send a scientist back to the drawing board, and usually does. After submission, there's usually some kind of change that the reviewers will require, including changes in actual experimental design. So you'll have to repeat experiments and things like that. But that's something that only makes the science better. After that, publication can happen after it's been accepted by experts in the field. And this process is known as peer review. It's a very important part of science, and something that you've probably heard a lot about. The other thing I just want to mention, since we did talk a little bit about repetition, is within an experiment you need many groups or subjects within an experimental group, but you also are going to have to be able to repeat the findings. So when you actually have those findings, they do need to be repeated by other groups, and oftentimes other labs are going to be repeating those experiments. And so your science is going to have to withstand that kind of test, and the most important science and important findings will withstand that kind of test. Okay, thank you.